Good evening, friends. So some of you requested me to create a video about the normalization of histology slides, that how Python or the computer programming can help in normalization. So here is a quick demo. This demonstration is based on this particular published paper that is titled as Color Transfer Between Images. It was published somewhere between 2021. Basically, if I summarize this paper, what this paper says is if we select a template image, the colors of the particular template image will be transformed onto the other sample images. So let's understand this concept by using some demonstration. I have uploaded the whole project on my GitHub page. I will share the link in the description. And if any one of you know how to replicate this, you can follow along with me. So this is the GitHub page of this project. Go to code, copy the link, open the folder where you want to replicate this project. I will open a terminal in this particular location and I will git clone and paste the link that we copied earlier. So it will create a new folder. Basically, it will replicate the entire project in my local directory. So here it is. Everything is replicated. So initially, we provide some input images to the program, and the output folder should be empty. On the GitHub page, I have uploaded the successful run project. That's why the output folder has the images. Initially, I will delete all the images since we will start from the beginning. So our output folder is blank. I have created two demonstrations, one with the natural images that has the simple examples of like sunset, autumn, ocean day, simple natural images I have selected. So what happens when we do the image transformation on these particular images? Later on, we will progress to the pathology images. So these are the sample pathology images that I have chosen to demonstrate. So now we have cloned this particular repo. I will open a terminal, and what I will do is in my computer, I will run a Jupyter Lab. So to run Jupyter Lab, I will first start my Conda. So I will type Conda activate. For just for the demonstration, I will use the base Conda only. So now my Conda is activated. I will start Jupyter Lab. And don't worry if you don't understand this particular lines. So you can just follow along with me if you are very new to this. So here is the project that I wanted to show you. You can also see, if you just want to see, you can just directly open the link which I have provided on the GitHub and go to this particular file, Reinhardt color transformation. Here I have demonstrated everything with the sample images. So if you just want to read this particular thing, you can read from the online also. Right now, I'm demonstrating by running each and every cell. So I'm deleting the output that I had generated earlier. I will go to kernel. I will clear all the output, restart kernel and clear all outputs. So now I have deleted everything. My output folder is blank. In my data folder, there are some natural images, and in the pathology images folder, there are some sample pathology images. So to run the program, first we need some important libraries that are required for this project. These are Numpy. This is basically in this particular project, if I talk, then we are using this to convert each and every pixel of the image into some numerical format. Later, the next important library is CV2 that is related to OpenCV. OpenCV deals with the pictures. So we need this particular library. Next library is OS, that is operating system library. So to run the commands of the operating system, we need this library. Last one is matplotlib. This one I am using just to show you the output images. It is not required for the project per se. Next, we have created a function that is calculating mean and standard deviation of any variable that is given as x. So it is calculating mean and standard deviation. These mean and standard deviation thing, I already mentioned that we are using from this particular research article. How we will use this mean and standard deviation, that I will come to, I will explain in the next few sections. So first, let's start with the natural images. So the first step would be to load the natural images directly. So it is in my computer, it is located in data, then natural images. So my input directory location is data, natural images. The next command is about creating a list of all the files that are in this particular folder. So I'm running this particular command. So if you see, there is an error because I forgot to run the initial section. So we will start from the top. First, we will load the important libraries. Next, we will run this particular function that we will use later on. The third one would be to load the natural images directory and create a list of the files. Next, we will set the output directory. The output images that will be generated from our program will be saved in the folder located in output, then natural images. Here, all the output files will reflect. So we have set the output directory. Next thing is we need to set a color template. So right now I have set color template, basically the template image as ocean sunset. We can select any of the image as our our template. For example, ocean day, ocean sunset, any of the image. We only need to be careful about selecting the template, because template would be the one the other images will look like. So for example, for this particular demonstration, I will select ocean day as the template instead of ocean sunset. So I will rename the file here ocean day. So now I am setting the template image. Next line is loading the image and converting it into BZR to lab format. Those who are familiar with OpenCV, they know that OpenCV uses BZR color coding. We are converting it to LAB. The last thing is we are using the get mean and standard deviation function that we ran earlier to calculate the mean and standard deviation of this particular image. So we have run this cell. Now comes the actual code. This is the only code that is using that we are using for image transformation, apart from setting up the template. So this code is the crux of this particular video demonstration. Let's see step by step what is going on. And here is a for loop. This for loop is using input image list. This, if you can recall, input image list that we have set, it was in the input directory, just below the input directory. What I mentioned earlier was input image list consists of the list of files that are there in the particular directory. Okay, so if you want, I can give you one demonstration here. Input image list, if I print it out, you can see it is a list that is containing the names of the files, all the images that are particular in that particular folder. So one by one, we are picking up an image, and after that, we are printing the image name. The next step is about reading that particular image. After that, we are using the same code. We are converting the BZR to LAB format. And lastly, same code, we are calculating the mean and standard deviation. So far, all the things are similar, which were there in the case of template. So where is the difference? The difference lies in the next for loop. Here, we are using the formula mentioned, which was derived from the description, detailed description given in this particular lecture article. So the formula looks like this. We have the input image. I mentioned Numpy is used to somehow convert every image format data into a numerical format. So from this image data, numerical data of the image, we are reducing the image mean data, and ultimately from the output, we are multiplying it by standard deviation of the template divided by the standard deviation of image, and ultimately we are adding the template mean. So that's the formula. That is the very important thing for this particular type of image transformation. Ultimately, we are recreating the whole image. So let's run this particular cell. It will take some time depending on the size of the images and the number of images. So far, it has converted 2 images. So if I go into our output, natural images, we can see that there is one image already has been listed. So now there are 2 images that have been converted. Let's convert it. After this, once this particular conversion is complete, we will see the comparison of the original versus modified image. Before seeing this comparison, let's first recall what was our template. It was ocean day. So everything should look like ocean day only. So it is completed. Now we move ahead for the next step for the comparison. Here it is. So now you see ocean day it is. So we use ocean day for the template. So as you can see, this is an autumn image. It is modified to some different type of color scheme that is looking much more like an ocean day. Here is the falling water image. The colors are changed for this one also. The sunset image has almost been converted into some like some different type of color scheme. So all of the images are converted into a different one using the template image. So the same concept we can apply for the histopath image or histology images. So here is a quick demonstration for the pathology images. We are again setting the input directory and also creating an input image list. So we have set the path output directory. Next, so the third one is we are setting up the template. Here I am setting up the template 1.png. You can choose any one that you want. Here it is. I have selected this particular image as a template image because I kind of feel like this all of the slides should have this particular intensity of HND. If you think other image should have been selected, you can try to do so. The program would work in each and every case. So here it has calculated the mean and standard deviation of the template image. Now the for loop, we will run the same for loop again. So it has started generating the output for the pathology images also. So it is finished. Now we will see the comparison. So there would be no change in the first image 1.png as we have selected it for the template one. There is a drastic change in 2.png, 3.png. If you can see here, in 2.png, eosinophilic and basophilic both colors were like faded out, while in case of after the processing of the image with the template, now they are looking like fresh slide. Similarly, in case of 3.png, we can say that colors are augmented. In case of 4.png and 5.png, the basophilic color is little bit suppressed. In case of 6.png, the eosinophilic color is suppressed, and similarly, the 7.png is also modified. So that's what I wanted to demonstrate. So this was a quick demonstration about this program. Let me know if you understand anything or if you have any doubt. You can ask me anytime. Thank you.